Good afternoon and welcome to Arizona Community Grand Rounds. We will begin in just a minute or so. We're letting folks gather. And we're anxious to move in today's presentation and conversation with Dr. Dave Engelthaler. So as folks join, we will begin in just a minute. Well, good afternoon, everyone, and welcome to February 2021's edition of Arizona Community Grand Rounds. My name is Mark Carroll. I'm Chief Medical Officer for Health Choice Arizona, part of Blue Cross Blue Shield of Arizona, and we are so lucky to co-host these rounds with the NARB Institute, and I'd like to note a special thanks to Mary Jo Gregory, President and CEO of the NARB Institute, for helping co-host, organize, and disseminate these rounds that we do once or twice a month based on topics that are of interest and availability of fabulous presenters. And we've been very fortunate to have fantastic presenters. Today is no exception. And I'll introduce Dr. Engelthaler in just a second. But just a reminder to folks that you have joined on mute. We do record these sessions, so they're viewable afterwards. But if you have questions during the session, at any time, please feel free to enter them into the Q&A, and we will aggregate your questions. And at the conclusion of Dr. Engelthaler's presentation, we'll go through as many of those questions and get his expert answers as we have time for today. Dr. Engelthaler's presentation slides will also be available after the presentation. So if you've missed something, don't worry, you'll be able to look at those slides and record or review the material later from the recording if you so choose. So let's dive right in. I'd like to introduce Dave Engelthaler. Dave is the director of TGen North, the Translation Genomics Research Institute in Arizona. Dave has been a colleague and friend of mine for many years now, and his bio is impressive, and I would not do it justice by trying to abbreviate it for today's introduction. But Dave is a trusted colleague, engaged community member, and leader in healthcare delivery, healthcare response, and I would call it next generation public health work in Arizona, the country, and really internationally. So without further ado, I'll turn it over to my friend and colleague, Dave Engelthaler, for today's presentation on the update in genomic epidemiology for coronavirus and COVID-19 in Arizona. Over to you, Dave.
Thanks, Mark. Totally thrilled to be here again and joining your grand rounds. The work that you do at Health Choice and Mary Jo's leadership at NARB Institute is it's definitely a blessing for Northern Arizona and for Arizona as a whole, and hopefully we're helping to carry on that great partnership as much as we can. I'm just honored to be part of it. And for everybody viewing today, I know a lot of you, I've talked with a lot of you and work with you on genomics as well as other things surrounding infectious diseases and epidemiology and pathogens. So I'm great to be seeing all of you again. Today I'm going to focus on COVID-19 genomics. Let me go ahead and start this presentation. That'll work better. COVID-19 genomics, and really with an emphasis on the variants and mutations. It's become kind of all the rage. It's, last year when we did grand rounds, that was, many months ago on genomics, we were really kind of describing how we're essentially sequencing the RNA out of these samples to understand the virus, and then building the family trees, and then understanding what's happening in Arizona, what lineages are coming in, what's moving around, and just getting a general sense of the pandemic. Now, it's really become one of the most important things that we can do is to identify and track these variants of concern. And so we've been doing that now for a while, and we have a good baseline of information now, I think, to share, and a baseline capacity in Arizona that's going to really help us see us through, I guess, this pandemic and track these variants along the way. And a lot of you have been following this very closely, much more closely than probably in the past. Not only was last year everybody became an epidemiologist, this year everybody's becoming a viral genomicist and is talking about these different strains and variants in a way that is, definitely at the subject matter expert level, and asking me questions that even I got to go back and look up sometimes the answers. So it's great. We're becoming more informed. I want to walk through a little bit of this. I only said all that because I know that a lot of you know background. So forgive some of the background. I'm not going to repeat what I did in the previous grand rounds, but I am going to talk a little bit about background to get us to where we are today. As everybody's really aware, we're able to develop these family trees of the virus, understand really how what strains are existing locally and globally, how the strains are moving around, because we can understand the relationship between each and every strain of the virus, or each and every isolate of the virus that we can get out of a patient. And really now what's become important is that some variants are really taking a foothold and changing the face of the pandemic and how we have to respond to the pandemic moving forward. And this is really becoming important now in the vaccine era. Not only are we looking at some of these variants that move much faster and move and can really dominate a population very quickly, some of them may have an ability to escape some levels of immunity. We'll talk a bit more about that, whether that immunity is from a previous infection. So we see some reinfection cases, very limited, but they have occurred, as well as then some post-vaccine cases, much more likely to start occurring now that there's been tens of millions of Americans vaccinated, getting close to 1 million Arizonans vaccinated. It's just very likely we will see some post-vaccine cases that essentially are escaping the vaccine, and we want to understand, is there something genomically in that? So that's what we're doing now. But let me just take one step back to last year, because this whole discussion around variants was occurring in certain circles last year, and I think it was somewhat overlooked. In fact, I've talked about it before too, and you may remember this spike 614G mutation. This was the mutation that we all talked about last year, and I was referring to it unofficially as COVID-20 because it really became the dominant strain in 2020. And just by looking at this particular tree, this is again the family tree of the virus. This is looking at thousands of strains from around the world as we march from the beginning of the pandemic really till about mid-year of last year. Just as a snapshot, what started to happen is that these gold-colored dots, these gold-colored leaves on the tree are the strains that had that mutation. This is essentially everything that showed up in Europe, ripped through Italy very early on, France, and then came to the Americas and dominated what we saw in the Americas. And then these teal-colored ones are the strains that were still originating out of China that didn't have this mutation, and they became less and less important around the world. And so I really do think that this mutation is the one that made the pandemic. It really was not, we did not see the strains that were coming out of Wuhan, China dominate the world. It was the one variant that was seen coming out of China, but through Europe and then into the Americas. We know that this mutation has been heavily studied now. This mutation in the spike protein actually changed the nature of the virus. It made it much more easy to infect human cells, replicate and transmit to other individuals, more efficient at transmitting between people, causing a higher level of viral load in those individuals. This was not the virus that was first seen in January and coming out of China of last year. This then became the COVID-20 virus. And then even in Arizona, we can see the same thing. dominantly almost everything we saw was this particular variant. And then there was a lot of subpopulations that came out of this variant, but they all had this mutation. And very few then of the teal colored leaves are essentially representatives of the virus that didn't have this mutation in Arizona. So now let's move into 2021, and I'll just call them, probably only on this slide, the COVID-21 variants, meaning that these are the ones that are now going to be the most important that we're following in 2021. The virus is evolving, picking up different mutations, and then some new variants come up that seem to be even more, what appear to be more supercharged than the variant we saw last year coming out of Europe. And the two that have been spoken most in the press and that we're following most closely in public health are the ones that originated out of the UK and out of South Africa. We're not supposed to call them the UK variant or the South African variant anymore. They do have official names, the UK variant being B1 or B11.7 or B117, and then the South African B1351. Now, of course, most humans don't talk like that, and I'm pretty hesitant to use those, but those are the official nomenclature. There's a new group that's actually taken to starting to call these by regionally important bird names. So you're not talking about the regional country, but maybe a bird from that country. Very interesting, and I'll talk about that. But there's a big argument about what we should call these variants. For now, I'll just try to keep it clear. We know what we're talking about. The one in the UK and the one in South Africa that are spreading to now multiple parts around the world. Both of these variants spread somewhere between 20 and 70% more rapidly, but no empirical evidence that they're more virulent or that the virulence has actually changed within these. Although we are seeing in the UK an increased number of asymptomatic cases as well as an increased number of serious cases, mostly because the overall caseload really took off. Now, the numbers are dropping, but what happened is that the vast majority of all cases became this one variant in the UK. The immunity from a previous COVID infection or from vaccine will prevent infection, or I should say, will prevent serious disease. However, the South African variant, that 1351, may evade some antibodies and some antibody treatment, so that the monoclonal antibodies specifically. And that's because there's been enough of a change in the virus, and I'll show you here that in a second, that some antibodies don't bind. However, the majority of antibodies that are generated by a previous infection or by vaccine still will bind and prevent serious infection, at least from what we understand so far. So if we just look structurally, I'm not going to really get too far into this, but just so you can visualize some of the differences and the similarities. What we're looking at here, this gray globby mass here, is essentially the spike protein. This is essentially what the conformation of the protein looks like when it's all the amino acids are folded together. And essentially what happens, the spike protein has essentially this tetrameric binding domain. So essentially allows for binding in 3 spots. So it's a replication of the protein. And then when you have a mutation, that mutation shows up in 3 different locations in this area. Now, what we're seeing between these 2 variants, they both have this same 501Y mutation in the spike protein, and that's in the receptor binding domain. And that is actually causing it to have a stronger, essentially, connection with the human ACE2 receptor, essentially the receptor on the human cell, and allows for quicker viral entry and replication, kind of like what the 614G mutation did. Really seems to be what's happening here. And so you get faster replication and then faster ability for that virus to get out of somebody into the next person. That's why we're seeing these kind of explosive outbreaks with this. Now, both of these variants seem to have that mutation. The other typical variant or mutation that's seen in the UK variant, if you will, is been the 69 to 70 deletion. The amino acids 69 and 70 are deleted. And that doesn't really seem to change the virus much. But what happened is that there were some assays out there, including the Thermo Fisher assay that some places have used, that it didn't work well when this mutation and this deletion showed up in the spike protein. Now, they look for things other than the spike protein that typically look at the N gene as well. So the assay overall wasn't seeing false negatives, but you were seeing a dropout of the S gene. And that's actually how we first found several of the cases, including some of the cases that we do now see in Arizona, is because this assay was no longer working appropriately. And then you go back and sequence and say, yes, the deletion was there and it is this particular strain. So it's become part of how we do surveillance to look for this strain. Now, this one here says UK variant on the bottom. That is not correct. I'm not sure how that happened, but that's supposed to say the South African variant. So my apologies there, and I'll fix that before I send the slides out. But this particular variant doesn't have that deletion, but what it has a little bit more concerning is this 484K mutation. Actually, it's E484K. It's been referred to as the EEK variant or the EEK mutation. We actually have seen it multiple times, and it comes and goes. It doesn't give a lot of, hasn't given a lot of advantage to the virus as far as increasing replication. However, it does seem to cause potentially some immune escape, some antibody escape, this mutation does. So you don't have as effective of an immune response overall, likely still to prevent serious infection, but not likely to completely evade antibodies when we do see it. The concern is that we'll see more of these, and then you'll get more immune erosion. Now, one more variant to talk about, and I apologize because that's really what this talk is today, is going to be a lot about variants, is the Brazil variant. So you'll see on the bottom, I just changed the name there. The official name is called P1. There's also P3, similar, coming out of Brazil. And we'll talk about where these variants are being found right now in a second. But what's interesting is that this Brazil variant seems to have these mutations as well. Maybe not moving and spreading as fast. It has its own set of mutations that might actually be, I don't know, slowing down or not really increasing the rate of replication or the rate of spread. But it's an important one for us to look at because it has that EEK mutation, if you will. Now let's look at, so I mentioned the Brazil variant. So there are other COVID-21 variants, and there will likely be more, because what's happening is that there's a lot of selective pressure to find variants that will move fast enough to continue to cause cases. As we have more and more community immunity, getting closer to quote unquote herd immunity, there's fewer and fewer opportunities for the virus to move unless the virus itself moves faster and has an ability to spread faster. And so that's why we're selecting for these fast moving variants right now. So more of these are going to pop up. And so the Brazil variant is one of those. And as I mentioned, it's got that 484K antibody escape mutation. And right now it's been seen in primarily in South America, a couple places in Asia and in Europe, not widely spread. I'll discuss that more in a second, but it has been seen in the US. More important in the US is one that is called Southern Cal, or Cal 20C. It seems to spread really fast in California. Again, I'll get back to this in a second. It has another mutation in the spike protein, of course, that looks like a possible escape mutation based off of mostly in silico data, really doing some analysis of the protein itself, that we think that there might be some antibody escape. Not a ton of empirical laboratory evidence to prove this yet, but we do think that this is possible. And it is becoming really important in California. Now, this is just a slide to show, this is kind of a grotesque looking slide. This is the bar through the middle here is essentially represents the whole genome and all of the different genes in the genome. The teal colored here is the spike gene. And what this is showing is all the mutations that are in those three important variants that we're tracking right now, the ones that we call UK, South Africa, and Brazil. So there's a lot of variants, or excuse me, a lot of mutations in just those three variants. It's only a few of those probably are important for changing the nature of the virus and the epidemiology of the virus. But they did come with a whole host of new mutations, which made it somewhat alarming. The thought is that each of these came out of an individual who would have been chronically infected, maybe immune suppressed, couldn't shed the infection, and allowing the virus to replicate over time, pick up more mutations. And so once they finally then exposed somebody else and infected somebody else, it came with a whole host of mutations that you normally may only pick up one mutation, maybe two during an infection. Here they were picking up dozens of mutations during a single infection. So it's an interesting thing to look at, but if you can pick up a lot more mutations, your chances of finding one that'll be selected for are increased. So we're just a side study, we're going to be looking more and more closely at chronically infected individuals to look to see if that is a common occurrence with these, and maybe we got to put a lot of extra care and caution to prevent transmission from those individuals. So a little bit deeper dive then into some of these variants or lineages. Again, here the B117 or the one originating from the UK. Across the top here, we can see again the genome and the different genes. And just a reminder, within this one, it's got that deletion, that 6970 deletion, as well as then the 501 mutation. But you can see there's other mutations in here, as I mentioned, a lot of other mutations that don't really seem to be changing the genome the virus all that much at this point. Additionally, what we're seeing is that it's become really increased prevalence around the world. So this graph here is just looking at the proportion of samples that are sequenced around the world that have this lineage, and you can see how important it's been. Now we've been overlooking for this one, so it's been very biased to look for these because we could see it with that one assay, if you remember. But what we see is 80%, upwards of 80% of genomes around the world are of this lineage. Now that doesn't mean it's not a strict representation around the world. It's just where people are sequencing: the UK, Europe, US, other places. This has become the dominant one in a number of areas. Not in the US, and I'll talk about that in a second. In fact, here are the numbers in the US. It's, if we go back to December, which is down here, it didn't even show up hardly at all on the radar. We couldn't really find too many older cases. But this is getting into January now, the end of January. This is getting close to 10% of all of our cases that we sequence in the US are of this lineage. So it is here, it is widespread, and you'll see the US data here in a second. But looking at the global data on the South African origin lineage or variant, we can see here, this is again the global data, and these are much lower percentages. So this is, we're looking at 1.5, 2%. And so we could see, coming into February now, it's still pretty low level. It has spread to other countries outside of South Africa. It is the most important virus strain right now in South Africa and other Southern African countries. But as it's gone into other parts of the globe, it has not really dominated yet. And as a reminder, it has this 501 Y mutation and that EEC mutation, that immune escape mutation. But then again, looking back at this particular graph, if we look in the US, we can see here that really we didn't start seeing it until sometime in January. There's some evidence that maybe it occurred late last year, but then we just could not find it until just recently. And it's been seen in a few states, and I'll talk about that in a second. And it represents about 2.5 to 3% of the actual sequences that have been looked at in the US so far. And then the Brazilian origin one is even spreading less. This is the one that's typified again by the EEC mutation, as well as then by the 501Y mutation that also shows up in there, like the South African one. Now here, when we're looking at well under 1% of genomes around the world. It's barely moved out of South Africa, or excuse me, out of South America, out of Brazil. But then when we look in the US, in the US, it's not even hardly showing up, and I'll show you why here in a second, because we've only seen it in 2 states. And then lastly, I wanted to show again the California lineage. This is the one that has that potential new escape variant mutation in it. We don't know for sure yet, but it's become really important in California. Just looking in the United States here alone, It is getting close to about 9% of all the genomes have this, are of this particular lineage throughout the US. And then in California, we can see even the last couple of weeks, this has now become about 60% of all the cases in California that are sequenced have this lineage. So it's really has taken a foothold there. And we are seeing it in Arizona. I'll show Arizona numbers here in a second. So now we've kind of moved from the global level to the US view. You can kind of see the progression here. The B117, the UK variant, now seen, this is yesterday's data, I don't have today's data in here. 1,100 some cases in the US have been identified and confirmed. We can see it in many states across the country. Looks like 40 states have reported a case, including Arizona. We've seen about 5 cases of this in Arizona. The 1351 out of South Africa, this is much rarer, obviously, in the US. We've seen it in 8 states, only 17 cases of this. So maybe, on average 2 per state where it's seen. It's not taken a foothold really completely yet. We don't know if it will spread rapidly. It doesn't seem to be a highly spreading as the UK variant at this point. And then the Brazilian one is been seen in 2 states in the US, Minnesota and Oklahoma. As of yesterday, a total of 3 cases. Very early. We're likely to see more cases of this as well. And we're looking for all of them. And so then that brings us to Arizona. And I want to just point out here that this is a dashboard. I'm just this little screenshot from a dashboard that was pulled together here. Haley on our team, who many of you know, had designed this, and we're trying to keep this up to date on an essentially daily basis as new genomes are produced, so that we can understand how much sequencing is occurring in Arizona, how much coverage across the state are we getting, also how many of the variants are we seeing, and then linking those to the different parts of the state where we see them, and then tracking our overall proportion. So on this state map, you would see, and I believe you see the link up here. You could actually see that live. You hover over this, you'll see the total number of genomes sequenced in each county, and then the proportion of the actual cases that have been sequenced. And then we're tracking that on a month-by-month basis, the proportion. This is actually a log scale here from 0.1 to 1 to 10%. Our goal is to get us at 5% across the board. I think we could potentially even get higher than that. We certainly can get higher than that with the months moving forward, but to get maybe an overall 5% of Arizona's cases, we'll have as good, if not better, than almost anywhere else in the country with these levels. This is genomes that are mostly have come out of TGen, but also being provided by multiple places, including the State Public Health Lab, straight from the Centers for Disease Control, Arizona State University, University of Arizona, and US Air Force and Quest Diagnostics, and now LabCorp as well. So multiple places are sequencing. We're hoping to try to get them into the thousands, all these groups together in the thousands per week. We're going to have a great handle on the genomics moving forward in Arizona. Now, just so you know that we're trying to stay on top of this. Anything that's coming up, we're tracking closely. A report just came out in medRxiv on Sunday about a new lineage, what's called 677Q, which is essentially, that's essentially the mutation in the spike protein. The lineage was actually named off of birds that are local, and this is seen mostly in New Mexico and then a separate sublineage in Louisiana. So they've been named quail and pelican, which is maybe easier to remember than the actual numbers that are assigned to these things. But just so we know, we had to go in, we looked in the database yesterday, we're able to identify, absolutely, we've seen this in Arizona. It's we have about 20 cases that we've at least sequenced recently over the past month and a half. This may become an important one. We don't know if it's being driven because that 677 position is really causing a change. These authors think that it may, because it's in the receptor binding domain again. So again, finding new variants that are being selected for, we're tracking them, and we're going to be tracking them in real time. Now, I kind of want to wrap up with just a couple of examples of how we're using this beyond just tracking variants, which is important, but the genomics gives us so much more information. And we can actually understand outbreaks and what's driving outbreaks and link it to epidemiology data to better understand what this virus is really doing in our communities. We have been sequencing, really since the beginning of the pandemic, and really done it. I think we've done a pretty good job of understanding what happened at the early stages of the pandemic. So the first 6 months or so, there was probably a gap in knowledge then the next 6 months where more limited sequencing was occurring. And now we're really ramping that back up again. But our goal is to put this in the hands of public health, not just so we can count variants and tell the media, but really so public health can do something with this information. And so we've reported on this, and I'll just show a little bit more information. We've been tracking a few different outbreaks to help out. One is in a tribal community where we saw that really essentially 2 different cases ended up causing the entirety of the outbreak across the tribe. The tribal population, and move really quickly. And they were their own variants. Now, we haven't really seen these variants outside of this area, which is interesting. It hasn't spread to other parts of the state. This is a fairly isolated area, but the virus did act differently. And we were able to use then the genomics to also track the actual case-by-case movement of the virus, using the on-ground epidemiologists, or working with the on-ground epidemiologists as well. So in this particular case, we did have a mutation in the spike protein, which we think was potentially changed the phenotype in a way that caused very rapid spread. What we were seeing in this particular population was 80, 90, 100% attack rates in congregate settings, in multi-generational homes, in correctional facilities. This thing moved very quickly and hit everybody, or nearly everybody, pretty fast, but also had a fairly low fatality rate in a highly comorbid population, and in a Native American population where we know we've really seen, unfortunately, increased severe effects of this virus. We're actually seeing much lower levels of fatalities. So this is probably a combination of great on-the-ground epidemiology and clinical action immediately, as well as the fact that the virus may be a little bit different. So we wanted to look at this virus more closely, this strain, and see, does this particular mutation show up anywhere else? Well, we're seeing on this is a global database of all these global cases. And this one gold leaf right here, this one case turns out to be the Arizona outbreak. We're not seeing it really anywhere else. We've seen that it's possible it's shown up in a couple of places and didn't cause outbreaks, but really this is an Arizona independent lineage that we were seeing. And then when we go in and do the genomics, we can build a family tree, but this isn't all that useful to public health just to see this. What does that mean? They all have that same variant. We can understand the population structure of the virus a little bit, but it doesn't really make sense unless you're working with the epidemiologists on the ground and tying that information together. And so this is information we're pulling together. Hopefully there'll be a manuscript out soon, but really what we're wanting to do is tie the epi data to the phylogenetic data so we can understand direction and source. We can understand what might be primary drivers of these outbreaks, and trying to provide it as quickly as possible so that we've actually turned the genomics into a very rapid turnaround process, which hopefully we can discuss here in a minute. So what the goal here then is to link these two together. And I'll just show an example of this, and probably some of you have seen this before, so I apologize. But on the left-hand side, that's really the family tree of the outbreak in this community. And then on the right-hand side, these are essentially the epi diagrams that link the cases together epidemiologically. And so in one particular cluster, this was the very beginning of the outbreak. There was a healthcare individual that actually had visited outside of the community, came back, became symptomatic, and continued to work. Unfortunately, it did not know at that point that they had COVID. And then all of a sudden, multiple colleagues of the healthcare worker became sick. The healthcare worker brought it back to their household. Colleagues then got sick, brought it back to their households, and then the outbreak goes from there. So it's the very beginning of an outbreak. What we did see of interest in this particular case was that they knew something was going on, particularly in household C. They were concerned for the kids, and this is very common. Get the kids out of the house, and they did. They sent kids to go stay in other households unfortunately, turns out the kids were asymptomatically infected, carried it to other households, infected those households as well. This is how these kind of outbreaks can occur in ways that we don't even appreciate until you can pull all that together. So what we wanted to do then was to tie the genomics into that. Does it make sense? Does what we think happened actually tie with the phylogenetics of the outbreak? And we were able to, when we zoom in on a portion of that, we can absolutely see the cases that are clustered phylogenetically absolutely were part of that initial cluster, and that was the beginning of the evolution of the outbreak. We had another cluster that involved a correctional facility and involved people who got incarcerated but previously then likely infected the household, which then ended up with other infections in the community. Again, we're able to tie that genomically as well together, and that makes sense. What we didn't know is the connection between these. We knew that genomically they're connected, epidemiologically we didn't. And by using the genomics, then we can actually find the individual that would be linking these two clusters together, and then try to retrace the steps on how that actually occurred when it didn't come up in the initial epidemiologic analysis. Okay, and then one more example, and this is a paper that is actually under review right now, that we're looking at an intellectually and developmentally disabled adult group home. And this is actually a set of homes, about 20 homes that are part of this system in an outbreak that started occurring. We can see on the right-hand side in this tree, everything that you see here in color was associated with that outbreak. And so what we had is in blue was staff that were affiliated, and then red that were residents that were part of the outbreak. And then in the black dots here, these are community members that weren't associated, but we saw the staff and the residents pretty much all had exactly the same strain. A couple of them showed up with one additional mutation, but it was a clonal outbreak affiliated with this residential home. There were a couple staff also that became infected that had nothing to do with the outbreak. They were infected somehow in the community. But what we want to do is also then trace it, how it moved through this set of this facility. And we could see in this particular case, the black dots represent staff, and then the pentagons represent different homes that are part of this overall facility. And these homes are spread out by a fairly good distance. And so what we saw was that staff that were infected were connected to multiple homes. And this was early on in May. And then 10 days later, we could actually start to see what was going on within those homes. Multiple residents would get infected, other staff would get infected, and residents would move between homes and move the infection from one home to the next. Once this was seen, public health was able to actually work with them to stop the movement of residents between homes. And then also, as it became more pronounced that staff had carried it to other homes, that staff would only be working with one home. In some cases, actually ended up co-living with the residents just for fear of taking it out to the community or bringing something new into the community or into the residents. And so they try to do a really good job. But even when you're so focused on this and trying to do a good job of not transmitting the virus, once you go in and do this type of analysis, we could actually see how the virus moved around. There's a full diagram. It's kind of almost like a spirograph. And actually, when you look at the genomics behind all of this, which I didn't show, it really does become pretty mind-bending and too difficult to go through here. But again, this is ways that we've brought the genomics together with the on-ground epidemiology to better understand these outbreaks. So with that, I'm going to wrap up talking about building 21st century public health. This is something that Mark and I and Mary Jo have talked about for years, that we really do need to bring these tools into public health and have been trying to do this before COVID, but COVID has become obviously beyond critical to do this. We can now test every case, we know that, but we really need to sequence every cluster. We need to have good genomic surveillance in the background, but we can sequence every cluster. We can sequence very large numbers of samples now and develop real COVID intel, and as well as for other pathogens. We need, though, also to train local public health epidemiologists. This data is not obvious necessarily by itself. We're trying to make it more obvious, but we need to train epidemiologists to use this data on a day-to-day basis and really build that future capacity. We need to use this data to determine causes of potential disease differences. If it moves faster, if it becomes more virulent, this data can help us do that. And so investigating these mutations and then the new lineage is a critical piece to this. Also, and this is really a TGen driven piece, but we're on the path now to create a 24-hour, $25 genome, really democratize this technology. It's been too difficult for public health departments to really incorporate sequencing on a day-to-day basis because it's been hundreds of dollars a genome, very difficult, laborious. We're trying to shrink that down so we can do it in our labs and then help translate it to other labs. So it's very cost effective. That is cheaper than doing a PCR assay. I do believe it's feasible that we can do this with creating whole genomes for these viruses. We're also trying, the other big hurdle is all the data that's generated, very difficult to work with in a hospital or even in a public health department, trying to automate the analysis. So once the sequence data comes off those great DNA sequencers, it automatically does analysis that's useful for public health and provides visualizations, automatically updates dashboards like I showed you, or some of the trees that we use. So democratizing the data. And one other pitch I want to make is that in Arizona, we're doing this. We need to do even more so across the country, is prioritizing bringing these advances to the native peoples of the Southwest. These are incredibly important parts of our population that always end up with the trickle down of innovation and gets out there late. We're trying to bring that early and use this to support our Native American partners as well. And then lastly, just thinking what we're doing is we're digitizing pathogens. If we can test it, we should digitize it so that we have that record of it, and we can do all sorts of analysis down the road. We can do some immediate analysis to have an impact, and it's now becoming technically feasible to do that. Got to give that shout out to our funding partners. You can see NARBA Institute up there, Blue Cross Blue Shield, a number of other partners in Arizona have really stepped forward and helped get this going. Now, hopefully, we're going to be getting some CDC dollars working with the state health department to really make all of this a true reality. We made the possibility a reality. Now let's make the possibility the reality moving forward. And there's my last slide. I've talked about genomic first responders in the past. It's another piece to the puzzle that TGen has certainly brought. And I get to be that face sometimes, but it's really all those other people that are doing all this work, an amazing small army of individuals in Arizona supporting Arizonans. And so I definitely want to give them the shout out for all the great work that they've done. And with that, Mark, I've used up the majority of the time, but I want to make sure I got through all those slides. So I'll turn it over to you for questions.
No, it's really important and very thorough and quite impressive work that you're doing. Hey, a shout out to all those Arizona genomic first responders there from the TGen team. We recognize that that Dave is your leader and voice, but we also recognize that you all are doing fantastic work and have worked way over time, typically 7 days a week during this pandemic. I should note, not just to advance genomic sequencing, we didn't even mention that TGen stood up a CLIA-approved lab for testing and has been actually doing a lot of testing and has been a key component of the testing across the state during this time. So we're really nice synergy of efforts, but a big shout out, big thank you to all of you from really the entire state. We so much appreciate it. So Dave, a bunch of questions have come in and you've generated a bunch of questions in me and Mary Jo. So I'll try in the next 15 minutes to encapsulate some of those questions. So the first one, talk a little bit, if you can, about terminology and names. Should any of us care that even if we've taken genetics in the past, that the letters and the numbers change? Is that meaningful for us to keep track of, other than being able to say the B117? Does that speak to a gene as well as then a particular amino acid on that gene? What are they referring to, the letters and numbers?
Even better yet, it really is completely arbitrary. But it was somebody who set it up at the beginning that we had major dominant lineages that were A and B, and then within that we started seeing sublineages. So you had 1, 2, and 3, and then within 1 you had a sublineage 1.1, and so these are the sublineages of sublineages. It's really I don't like the new nomenclature. I know it's necessary for us to use it so we can keep them all separate, because there literally are hundreds of thousands of lineages because of the way that the virus mutates. The ones that are important we have to track, and we got to make it easier for our colleagues as well as the general public to better understand this.
Well, thanks for mentioning that. I didn't know that. I was actually trying to look it up over the weekend and figure out how these things got lettered and named. So that's actually helpful for me. It was really interesting just to see some of the information you shared in terms of what percent over time we've been able to identify that some of these variants are here in the US, let alone around the world. What's the data look like from around the world? Do we have gaps in where we're receiving information? Someone asked a question about data from China, and I know there's been an international team of epidemiologists working there. Are we expecting to round out more information from around the world, or some of that going to be influenced by factors outside of science?
Yeah, we're definitely going to continue to see, we have major gaps. There's some serious black holes in around the world where we don't get any data. And interestingly enough, I don't think there's still been a single case identified in North Korea. So that's, I find remarkable. But that's because of the local governments in different places. In some cases are either not doing testing or they're not doing sequencing. And maybe in a lot of cases, because they don't have the capability, they don't have the national infrastructure. I actually was just on a 2-hour call this morning with a national group that is really trying to work with the CDC and major foundations to really improve the fact that we have gaps across the US. We have a lot of black holes where some states haven't been doing hardly any sequencing at all. Some states have been lucky enough to have partners that can do a bit of sequencing, and so have been really kind of filling in that knowledge. But even within Arizona, where we're fairly robust, we have gaps in time and in some spaces as well. So it's a lot of that is a really important part of what we're talking about now. For surveillance purposes, we got to fill in those gaps of our knowledge while maintaining high level of sequencing, at least in key areas that can do it.
Do you think our terminology is going to revert a little bit to SARS-CoV-2, given that now there's COVID-19, COVID-20, COVID-21 variants? Not that that's important, but over time, we're going to have to settle on some way to refer to this emerging group of variations in the SARS-CoV-2 virus. Do you think that's meaningful? And I guess I'd like to also defend the poor quail and pelicans that that may be named for the variation in certain states through no fault of their own. They did not transmit this virus. They're being identified.
Exactly, and that's the kind of trap that we kind of fall into sometimes. Is we can find reasons to use or not use different types of naming schemes. Going back to the original part of that question, everything that comes from this pandemic and everything that happens in the ensuing years, those will all still be SARS-CoV-2 viruses. This is the virus. That is the name of the virus. Then you have all of these lineages. And so in these lineages, which essentially are on that tree, are different branches. And they could be some of the really long branches or some of the shorter branches. There's not a great defining line about what's a lineage and what's a variant and what's a strain. And there's no reason to really get bogged down in that. I think what we do try to come around and get to is what is the most important thing for everybody to know now. And right now, pointing out those different variants, those are the ones that are most important. We're going to probably see more in the coming months that are going to pop up because for some reason they're being selected for, most likely through immune escape. That's what it's going to be selected for here in the future. So we're going to call this all SARS-CoV-2, and then we're going to have lineages we talk about now and totally forget about in 6 months because something else is going to come up.
And has this virus, have the mutations this virus has had and the variations, the lineages, have those been more or less scientifically predictable based on what we know about the virus and what viruses historically will do, just for their or how the evolutionary pressure on a virus may exist? Are we likely to see some new types of variations in other sectors of the virus that we haven't seen yet that will be meaningful?
Yeah, great question. One thing the one thing that is predictable is that the virus will continue to mutate, and that some of those mutations will provide some type of an advantage that'll be selected for. The state of the pandemic has changed. We had at the beginning, everybody was susceptible, and so selection for things that really increased transmission wasn't as strong as it has been now, where you have 30-40% immunity in a population because of previous infection, because of vaccine. It's harder for the virus to move to the next person. So we are selecting for that now. And then soon it'll be selecting for things that actually can escape the immunity. So that, I think that progression is somewhat predictable. The actual mutations that will be selected for are not necessarily, although we do know that most of the concern here is going to be the mutations in the spike protein. There will be mutations in some of the other genes that likely will help offset any negative effects that occur from some of the spike protein mutations, so they'll be important. But we haven't seen selection based off of any of those other mutations at this point, because the first and foremost thing that's most important in this virus is how it connects to a human cell, and it does that through the spike gene.
So could you chat a little bit more about what we know historically in terms of viruses, contagious illnesses, and evolutionary pressures and selection? Do they tend to select towards a virus which transmits easier but is less virulent for obvious reasons? Do we know this? Is that just a theory? In other words, how worried should we be that there will be new mutations that will become more virulent and become the more dominant strain based on what we know historically?
Yeah, Mark, I think the first example you gave is pretty much the common hypothesis about what is the general evolutionary strategy. For coronaviruses, what we do know, the only successful strategy for coronaviruses to become human adapted has been to become more easily transmitted, but less virulent, which is why the four that are well adapted to us cause nothing more really than the common cold. Occasionally you get some outliers, but that's been the successful strategy. My guess is that will be the successful strategy for this coronavirus. SARS went away. It did not develop that strategy. MERS, another one that we get, the Middle Eastern Respiratory Syndrome virus, is still hanging around, but it's not hanging around in humans. This is not really an important human disease. It does cause cases and little mini outbreaks, but by and large, it's not even on the radar screen. So I do think that is the likely direction for this. It's not the case with all viruses. Rabies virus has not become less virulent. It has had to maintain a high degree of virulence because strategically that's what it's needed to help perpetuate the spread from mammal to mammal. So it's not always the strategy, but it is in a large number of cases, and I think that's the case for coronaviruses.
So we obviously have more to learn, but we also can base some of this on other coronavirus experience that we've had, which is important and perhaps somewhat reassuring. It's been a rough 12 months. But that leads to the testing and vaccination question. There have been some comments and questions around do variants, how do they affect testing, whether that be PCR or antigen type testing? Can you answer that?
Sure. Well, the one example that I gave earlier about the dropout for the S gene assays, for some of those assays because of that deletion that was seen in the UK variant, that actually did affect the test, and it was causing essentially discordance with the other assays or the other genes that were part of that test. But then it ended up becoming part of the strategy, or at least the surveillance strategy. Not that everything that has that deletion really ended up being the UK strain. It wasn't, but it still was a good indicator. but none of the other, no assays that we know of completely aren't working because of any of these variants, any of the PCR assays. Then the question is, what about the rapid antigen tests as well? Well, the vast majority of those are based off of the N gene, which is the nucleocapsid antigen. And right now, it has been tested for the UK variant, not affected at all. And there is no indication or likelihood that the other strains will be able to escape those assays. So we do anticipate that the antigen assays will continue to work correctly moving forward, but we got to keep eyes on it. We got to keep testing it when new important lineages pop up.
And we have some therapies that, while not perfect, are useful and effective. For example, monoclonal antibodies. So big plug for all my colleagues right now, from me, but I'm sure from Mary Jo and Dave as well. Please keep in mind that monoclonal antibodies actually are effective treatments early in a course of COVID for the eligible population as per criteria. Even with very mild symptoms, they actually not just prevent hospitalizations, but can save lives. But what about those monoclonals, Dave? Will we see new monoclonal antibodies, laboratory developed antibody therapy that will take into consideration the variants?
Certainly, and we know that they're at risk because monoclonal antibodies are, by definition, are going to be targeting one particular region of essentially the spike protein. And so they could be at risk. And we do know that some monoclonals looked at in South Africa have been having some escape problems by the variants that are there and the monoclonals they're using. Right now, it doesn't seem to be we're not at risk for what we're using primarily here in the US, but we could be. So we got to keep watching that very closely. And more monoclonals are being developed. I can even say our group at TGen, working with the City of Hope, they've recently have identified and are developing a new monoclonal as well that could potentially be used for therapy. We know lots of groups are doing this. So yeah, we got to stay on top of that because the virus will continue to evolve. Same thing with the vaccines, right?
Please speak about vaccine.
Yeah, right now, the vaccine makers have been testing against these variants. The ones the vaccines we have right now, Moderna and Pfizer, seem to still have as high effectiveness against all of these, even though there might be some immune erosion. It won't really change the overall effectiveness of the vaccine right now. What does look interesting is the AstraZeneca vaccine in South Africa. They are seeing some problems to the point that the government actually stopped the use of the vaccine there for that particular variant. But all vaccine makers are on record. They're going back. They're developing their plans for additional formulations to be able to handle these and potential new variants that might have immune escape in the future. Right now, it looks like the vaccines we're using are still going to give you enough protection to certainly prevent death and prevent serious disease, and by and large, prevent overall infection.
I think that's a really important point. 100% efficacy or lack thereof does not mean not even partial effectiveness can be actually very helpful for an individual as well as communities. Well, I know that you have and Mary Jo have championed this conversation more broadly before COVID-19 around One Health and around democratizing, as you said, the ability to do genomics. And I liked your phrase, digitizing pathogens. Any closing thoughts from either you or Mary Jo in terms of where we might go in the future to look at what a next-gen public health system looks like? And Mary Jo, I think we have a topic for future grand rounds to bring in some of our public health leaders and have that conversation. But Dave, any thoughts about that?
Sure, just real quickly, we in the community, but TGen also specifically have been working with NARBA and others to really start to bring these tools and technology into public health early on before COVID. But now we're really putting in full scale operation and seeing what it can do and what we can do. We're going to be even better post-COVID. We should be thinking about how we do this now for every critical pathogen. I think we're at that place now. We can really be in 21st century public health.
Thank you. Mary Jo, any closing comments? I monopolized all the questions. I'm sorry.
No, I would agree 100%, and I'm glad you highlighted it, David, and at the beginning and at the end about the 21st century public health. I think that is really where we need to go. And this has been incredibly informative, but I think we're only at the beginning of that. And the more awareness we can get out there on all areas, both the very scientific components as well as the practical components, it's where we need to be. And I like the $25 genomic, the idea in there. I think there's great opportunities. So I'm very grateful, Dave. I know you're busy. There's very few minutes you have free, and to put together this profound presentation, informative to even the most scientific of us and those that are on not in that but really want to learn this science. It's been wonderful, and we hope that we can impose on you again as this continues. Thank you very much. And to everybody, we had 99 participants, I think. So appreciate everybody's time on this very important topic.
And with that, we'll close. Thanks, Dr. Engelthaler. Thank you, Mary Jo, Gregory. Thanks to all of you for participating. We'll continue with Arizona Community Grand Rounds. Thanks for all the great work you continue to do. Please remember, kindness spreads faster than any virus ever will. So thank you for spreading kindness during sometimes very challenging months and years. Take care, and we'll talk soon.
Thank you, everybody.
Thanks again, Dave.